Hello everyone and welcome to today's Crush the MCAT lesson on enzymes and enzyme regulation. Enzymes are proteins that bind substrates in their active site, catalyze a reaction, and then create and release a product. We can see an example of this happening on the right-hand side. Catalysis increases the rate of reactions by lowering activation energy. Importantly, enzymes are not consumed when they catalyze reactions. There are two models you need to know describing how enzymes bind substrates. The first is the active site model, also called the lock and key model. This is when a substrate perfectly fits an enzyme, and we can see that in the example image here. The second is the induced fit model, and I have this bolded because you're more likely to see it on the MCAT. This is when an enzyme conforms when a substrate binds. Let me draw an example for you now. Here's my example, and you can see how the enzyme slightly changes its shape so that the substrate can more easily bind it. A couple more things you should know: enzymes work best around physiological pH and temperature. High temperatures in general increase reaction rates, but eventually a high temperature will cause protein denaturation, and when an enzyme undergoes denaturation, it'll stop working. Any pH deviations will lower enzyme function. Remember, different enzymes have different ideal environments, whether it's the acidic environment of the stomach or the more neutral environment of the small intestine, but if you become too acidic or too basic compared to their ideal baseline, you're going to lower the enzyme function. Enzymes might need additional parts to function. Coenzymes are proteins, and they're usually organic, like NAD+ or FAD. Vitamins are often coenzymes, or they're precursors to coenzymes. Cofactors are non-proteins, usually they're metal ions or they could be small molecules otherwise, and if a cofactor is tightly bound, it's called a prosthetic group. Some terms you may see are apoenzyme, which is an inactive enzyme without its cofactor. When you add that cofactor in, you create a holoenzyme, which is an active enzyme with its cofactor. Zymogens, or proenzymes, are inactive precursors of enzymes that require some sort of modification to become active enzymes. An example you'll often see is digestive enzymes from the pancreas. Those only become active once they are in the small intestine. Once they reach the small intestine, they're cleaved by other enzymes, and then they will start to digest proteins. If this mechanism wasn't in place and the pancreas stored large amounts of active digestive enzymes, it would end up digesting itself, or it would take a large amount of energy to prevent the pancreas from digesting itself. So we have this very clever design where we can store large amounts of digestive enzymes and deliver them en masse to the small intestine so that we can easily digest large meals. You should know all of these types of enzymes. Isomerases make isomers, ligases join two molecules together, transferases transfer functional groups, lyases break molecules and form double bonds, hydrolases break molecules with water, and oxidoreductases transfer electrons with oxidation and reduction reactions. Some enzymes have allosteric sites, which are separate from the active site, and those are where activators or inhibitors can bind to increase or decrease enzyme activity. Cooperative enzymes have multiple binding sites and bind substrates differently based on how much substrate is already bound. A classic example of this is hemoglobin. While hemoglobin isn't an enzyme, it is a protein which shows positive cooperativity. Positive cooperativity leads to a sigmoidal curve. If we look at the curve on the right here, we can see initially the curve is quite shallow. If I was to draw a tangent line at the bottom of the slope here, it would be quite shallow. However, as we bind more and more oxygen here on this hemoglobin curve, we can steadily see this slope increasing. Once we get to this point, the slope is at its steepest, and then we can start to see the slope leveling off. The reason it levels off is because the hemoglobin eventually gets saturated and you start running out of binding sites. Make sure you remember this association with positive cooperativity and the sigmoidal curve. Hill coefficients show whether a molecule is cooperative. In that case, they'll be greater than 1. Not cooperative, in that case, it'll be equal to 1. Or negatively cooperative, In that case, it'll be less than 1. Negative feedback causes the product of a pathway to lower the flux of the pathway. If we look at this diagram on the bottom right here, we can see that with negative feedback, we will have some kind of input, which will increase the output, and then as the output is increased, it'll negatively feedback on the input to lower it, and then as the input is lowered, eventually the output will be lowered too. Some examples of this are insulin release. High blood sugar causes the release of insulin, and the release of insulin lowers blood sugar. As blood sugar gets lower, the insulin demand gets lower too, and insulin will eventually stop being released. Sweating is also an example of negative feedback. The input of a high body temperature triggers the release of sweat, and sweat will lower your body temperature with evaporative cooling. As your body temperature becomes lower, the demand for sweat also becomes lower, and eventually you'll stop sweating. Positive feedback causes the product of a pathway to increase the flux of the pathway. A classic example you'll see of this is labor. So oxytocin is a hormone which strengthens uterine contractions during birth. Contractions push the baby downward through the vaginal canal and stretch the cervix. An increased cervical stretch increases oxytocin generation. So the input here is the cervical stretch. As the cervical stretch increases, we go through the pathway and we increase the output of oxytocin. The increased output of oxytocin will actually cause more cervical stretch because it helps the baby get pushed downward through the vaginal canal. That will release more oxytocin, and this path will keep repeating until the baby is delivered. And that's what will end this positive feedback loop. Feed-forward loops anticipate change and prepare the body for it. A classic example of this is salivation. When we smell food, we anticipate that we're going to eat it, so we start salivating, which will help with digesting that food. Michaelis-Menten plots are particularly high yield for the MCAT. A Michaelis-Menten plot measures reaction velocity against substrate concentration. The reaction velocity or reaction rate is the speed of a reaction and the formation of products. Vmax is the maximum reaction rate with full saturation of all enzyme active sites. Km is the substrate concentration at which half of Vmax happens. Important to understand here, Km is a substrate concentration. So while Vmax might be in units of 10 molar per second, Km is going to have units of, let's say, 2 molar. Kcat is another term you'll see. This is the turnover number, and it describes the amount of substrates changed per enzyme per second. Two equations you should know. Vmax equals enzyme concentration times Kcat. This is given in units of molar per second. And catalytic efficiency, which is equal to Kcat over Km. You may also see this equation here. What this equation is describing is the velocity of a particular reaction given the Vmax and the Km and the particular substrate concentration you have. So you could have a substrate concentration of 1 molar or 3 molar and so on. It's not particularly high yield to memorize this equation, but it is high yield to understand the general trend of Michaelis-Menten curves, which is to say that as you increase substrate concentration, reaction rate will go up. You should understand that at Km, this is half of Vmax, and as you add more substrate, you will start to approach Vmax, but you will never quite reach Vmax, which is why we can still see this gap here. There are a few types of inhibition you should know for the MCAT, and these are all high yield. If we take a look at the top right here, I want to first show you normal catalysis. An enzyme noted E here will bind with a substrate S. This will make the enzyme-substrate complex, which undergoes catalysis to make an enzyme with a product. In competitive inhibition, the inhibitor binds to the active site. This is normally where the substrate will bind. This does not change Vmax since the enzymes themselves aren't affected. If you have some competitive inhibitors floating around in a solution, but you still have enough substrate to make the maximum number of enzyme substrate complexes, you will still have the maximum reaction velocity. However, because those competitive inhibitors are competing for the active site with a substrate, this will increase Km. Remember that Km is the substrate concentration at which half of Vmax happens. So if you have competitive inhibitors fighting substrate for active site, you'll need more substrate in the solution to fill 50% of those active sites and get half of Vmax. These sentences can both be summed up by saying competitive inhibition can be overcome with increased substrate concentration. Uncompetitive inhibition is when the inhibitor only binds the enzyme substrate complex. So uncompetitive inhibition is the complete opposite of competitive inhibition. The inhibitor only binds to the enzyme substrate complex after the substrate is already bound. This will decrease Vmax since less enzymes become active in the solution. When the inhibitor binds, an enzyme, as we can see here, is actually knocked out of the equation. And so if your Vmax relied on 100 out of 100 enzymes being present in the solution and all working at top speed to get the maximum reaction velocity, then when you add an uncompetitive inhibitor, you'll drop this to 99 enzymes active in the solution. In this hypothetical example, you'll only reach 99% of Vmax no matter how much substrate you add. Km will actually decrease with uncompetitive inhibition, and this is due to Le Chatelier's principle. Briefly explained, because you are driving down the amount of enzyme substrate complex in the solution, you will drive the reaction to occur more in that direction. Because you're driving the reaction to occur more in this direction, it'll be easier for the enzyme and the substrate to bind together, and that will decrease Km. Mixed inhibition is when the inhibitor binds to the enzyme or the enzyme substrate complex. You'll notice that when we look here and here, mixed inhibition and non-competitive inhibition look pretty identical. And that's because non-competitive inhibition occurs when the preference of the inhibitor binding to the enzyme or the enzyme substrate complex is 50-50. In all forms of mixed inhibition, Vmax will decrease, again because less enzymes become available. You're knocking out this enzyme substrate complex from fully finishing the reaction. And in non-competitive inhibition, when this is 50-50, Km will actually stay the same. If we think about Le Chatelier's principle again, if we're knocking out the ability of the enzyme to undergo the reaction in this direction, and we're also knocking it out in this direction, then we're not driving the reaction to go either to the left or to the right, and so Km will stay the same because of that reason. I recommend to master understanding these inhibitions, you write out this statement here, and then you edit it to include all these types of inhibitions, and you really work to understand why this changes Vmax and Km. Once you understand why, you'll always be able to reason it out, and you won't have to rely on memorizing, which can be very difficult to rely on once you get nervous when you take your MCAT. Okay, I've summarized what we just talked about here. For competitive inhibition, remember the active site is affected. Competitive inhibition increases Km and it does not change Vmax. With uncompetitive inhibition, the enzyme substrate complex is affected. Both Km and Vmax decrease. And for non-competitive inhibition, you can either affect the enzyme or the enzyme substrate complex. You'll have no change in Km and you'll decrease Vmax. Again, work with this equation, work with this table here, and work with everything we talked about on the last slide too to make sure that you master these types of inhibition. Okay, you should also know about Lineweaver-Burk plots. These are the linear versions of Michaelis-Menten plots, and they're plotted with a few values that you should know. The x-axis is comprised of 1 over the substrate, and the x-intercept, which we can see here intercepting the x-axis, is negative 1 over km. So we can see that here or here. One thing that may be difficult to understand intuitively is which way these lines go when you have a bigger number. Whenever you struggle, I recommend plugging in extreme numbers and seeing which way those will go. For instance, if we have a graph here and I plot 1 over 2, let's say I'm right here. If I plot 1 over 999, that number is so close to 0 it might as well be 0. And so when I plot that number, I'll be over here. With this on a Lineweaver-Burk plot, I can see that if I increase the substrate concentration, so if I go from 1 over 2 to 1 over 999, that I will become closer to 0. And it's the same thing with the x-intercept. Don't let the negative sign trick you. If I have negative 1 over 2, it's the same. I'll just be over here. And if I increase Km, and I bring us to negative 1 over 999, again, I'm basically going to be at 0. So the bigger number you have when it's on the bottom, the closer to 0 you'll be. The y-axis on a Lineweaver-Burk plot is 1 over v, and the y-intercept is 1 over v max. Again, if we have a large v max, if I were to draw a graph here again, if I have a large Vmax, let's say 999, I will basically be at 0. And if I decrease the Vmax somehow, let's say to 1 over 2, I'll be further away from 0, I'll be higher up, and I'll be here. With all that in mind, let's briefly talk about the types of inhibition and how they look on a Lineweaver-Burk plot. So when we look at competitive inhibition, remember Vmax does not change, so the y-intercept will stay the same. The blue line here we have is the control line, and the red line is what happens when we add the inhibitor. So for competitive inhibition, Vmax will stay the same. And when we look at Km, the x-intercept, negative 1 over Km, gets closer to 0, which means the Km term becomes a bigger number. When we look at uncompetitive inhibition, we see that the y-intercept is farther away from 0, and therefore the Vmax term in the denominator must be a smaller number. So Vmax has decreased, and if we check Km, the x-intercept isn't shown on this graph, but if we were to extend this line, we could see that it's farther away from 0. Again, because it's farther away from 0, we know the denominator term is smaller. Therefore, Km has decreased here as well. And then for non-competitive inhibition, we can see here the x-intercept is the same, so we know that Km is the same, and the y-intercept has gotten farther away from 0. Just like for uncompetitive inhibition, again, this means that the Vmax has gone down. Keep practicing these until you truly understand them, and don't be afraid when you do your first questions involving Lineweaver-Burk plots to take it very slow and make sure you really test your understanding. Let's look at a couple more charts demonstrating the different types of inhibition. Here we can see Michaelis-Menten plots. Again, we see with competitive inhibition, Km is increased, which means the Km will become farther out, but Vmax is the same, so both of these curves will approach the same limit. For uncompetitive inhibition, we have a lower Vmax, so the curve approaches a lower limit, and also a lower Km, which means that we will get our Km faster when we have an uncompetitive inhibitor. For non-competitive inhibition, again we can see that Vmax is decreased, so the curve approaches a lower limit, but Km is actually the same. These are also demonstrated here in these Lineweaver-Burk plots. And finally, this slide puts it all together so you can compare the Michaelis-Menten plots and the Lineweaver-Burk plots with the different types of inhibition. And that concludes our lesson on enzymes and enzyme regulation.